All right, ladies and gentlemen, for this presentation, we're going to look into the exceptions to Mendel's rules. So all the things that we've discussed so far, dominance, recessiveness, the way that we look at the probability we get from the outcomes in Punnett squares, what we're actually doing is we're laying the foundation from which we can then understand the way things work. What we've learned so far is what we call simple dominance, and that's where one trait biochemically is dominant and can hide another. That isn't actually that common in nature. Instead, we have a whole host of other ways that dominance can play out. And so what we want to do is take a look at a couple of these examples to make sure that we understand that there are alternate ways for our genetics to create the phenotypes that we see. So the first one we want to look at is what's known as incomplete dominance. Now, incomplete dominance is known mostly for a blending phenotype. So typically for a monohybrid cross that you would see, like is in this picture, you would have two phenotypes. But when we have incomplete dominance, there is always three phenotypes. There is the dominant phenotype, there is the recessive phenotype, and now there is a hybrid phenotype. So this is a red snapdragon, which is a plant where if you are homozygous dominant, like the example up top, you appear to be red. That's your phenotype. If you are homozygous recessive, and they're using a different little symbol here, but we could put little r and little r, you appear to be one way, which is white. And what you'll see in here is that all of these are the heterozygous genotype. But because this is incomplete dominance, in this case, heterozygous is a new phenotype that is a blending of the other two. And in this case, that blending appears to be pink. So that's incomplete dominance, where you end up with a third phenotype that's actually a blending of the original dominant and recessive phenotype. Now, another thing that I want to talk about is what we call sex-linked traits. So, you have 46 chromosomes, all right? And those 46 chromosomes come in 23 pairs. Well, 22 of them are roughly identical matches. Actually, let's not use identical. They are similar matches. All right, so they're roughly the same size, the same shape, and they have the same genes on them. Now, they can have different alleles, different versions of those genes, but they have the same genes. But the thing is, that's 22. The last pair are known as your sex chromosomes. And they're important because if you're a female, they're just XX, and it's no big deal. They're the same as your other 22 chromosomes. But if you're a male, they are X in what we call the Y chromosome, which is actually much smaller and has much less genetic information on it. So let's just look at a basic sex chromosome, right? So here's a male, X and Y. Here's a female, X and X. And so what you'll notice is, if you do a normal Punnett square, this potential offspring is female, this potential offspring is male, this potential offspring is female, and this potential offspring is male. Great. Awesome. No problem. The thing about this is, though, that when it comes to traits, you have some genes on your sex chromosomes that are related to something other than just whether you're male or female. And so that can create some interesting dynamics when it comes to how those traits are passed on. Okay. So one of the things that's really important to understand is that the Y chromosome doesn't really hold traits. And this creates a problem for males because males only have one copy, one allele of several traits, because the Y chromosome, which they received from their father, didn't hold any traits, but the X chromosome did. And so what this does is this creates unique outcomes based on gender. So let's take a look at, we'll do hemophilia, because the letter H is an easy one to use. So hemophilia is a disease where your blood doesn't create clotting factors, or the things that lead to scabs, the way it should. And so people who are hemophiliacs, their blood doesn't really clot very well. So for them, a paper cut could bleed for days. You know, a small cut on your arm that you get like playing a sport or like running around outside, they could have to go to the hospital and get stitches because it won't stop bleeding, and that's a big problem. Well, hemophilia, the trait, the genes for this are on the sex chromosome. So hemophilia is a simple dominance situation where if you are big H, you have normal blood. If you are big H, you have normal blood, but you carry the trait. And if you are little h, you have hemophilia, and you don't really make scabs right. Okay. Now, what makes this unique is that the H is on the X chromosome. So if you're a female, right, you're XX, you're XX, you're XX. But if you're a female, you can carry a big H and a big H, one on each of your X chromosomes. You could carry a big H and a little h, one on each of your X chromosomes. Or you could carry a little h and a little h, one on each of your X chromosomes. But if you're a male, you are XY, XY, XY. And what happens is, for a male, you carry a big H and nothing is brought on your X chromosome. Or you carry a little h and nothing is brought on your Y chromosome. And this creates a problem because males can't carry it. They can't hide it. So their chance of ending up with the disorder is actually at a higher rate than that for females. Let's take a look at this as an example. Right? Let's say that I have a female who has normal blood but carries hemophilia. And then over here I have a male who has normal blood, and you'll notice I don't write anything over the Y chromosome, and that's because the Y chromosome just doesn't have the genetic information for this trait. And so what this does is this creates a female outcome that is homozygous dominant. It creates a female outcome that is heterozygous. It creates a male outcome that is phenotypically normal blood, because it's only one allele, so I wouldn't use terms like homozygous and heterozygous. And it creates a male outcome that is phenotypically a hemophiliac. And this is what you see. Right here, males have a greater chance of ending up with the disorder because they have no way to hide it, since they have only one copy of the allele. And so what this means is that you get different probabilities based on gender. Okay? And that's the real key when it comes to sex-linked traits. Because that Y chromosome doesn't really carry genetic information other than for gender, males end up having increased probabilities for certain disorders. Hemophilia is one example. Red-green color blindness is actually another example. It's far more common in males because they don't have a Y chromosome, or their Y chromosome doesn't carry any genetic material to help hide it. All right? So that's sex-linked traits. The last example we want to talk about is something called codominance. Now, when it comes to codominance, please don't misunderstand. This is going to seem very similar to incomplete dominance. But codominance is not a blend. All right? It is not a blend of the traits. Instead, both traits are expressed fully. Okay, so here's an example with chickens. All right, so you see over here, now they're using two letters, which can be confusing, but it's acceptable. All right, so over here we have white chickens, and white chickens are homozygous for the white trait. And then up here we have a black chicken. Black chickens are homozygous for the black trait. When you breed those two together, every chicken offspring they create will have one allele for the black trait and one allele for the white trait. Well, this isn't simple dominance, so one of them doesn't end up hidden. This is not incomplete dominance, so you don't end up with gray chickens because it's not a blending of white and black. Instead, you end up with chickens that have a checkered feather pattern, some white, some black, because what you're seeing is both traits are expressed fully. Now, this can create a situation with multiple phenotypes. Let's take a look at an example that's more closely related to humans. So here you have human blood type. There are four main human blood types, A, B, AB, and O. All right. Now, each one of these blood types is a co-dominant expression of alleles. Now, there are actually three alleles for blood. There is the A allele, which puts an A protein on the surface of your red blood cells. This is related to red blood cells, okay? There is the B allele, which puts a B protein on the surface of your red blood cells. And there is the O allele, which puts no B and no A on the surface of your red blood cells. So what this means is you could have one copy of the A allele and one copy of the O allele and have A-type blood. You could have two copies of the A allele, like right here, and have A-type blood. You could have two copies of the B allele and have B-type blood. You could have one copy of the B allele and one copy of the O allele and have B-type blood. But if you have one copy of the A allele and one copy of the B allele, since this is co-dominant, meaning all of the alleles are expressed fully, on your red blood cells you'll have A proteins and B proteins. And that happens because it's co-dominance. And what this means is, even though when you have AO as your genotype, O is fully expressed because O just means no B and no A. But the A is fully expressed because it's putting A. So the only way to end up with O blood is to end up with two copies of the O allele. And that's why there's such a variety within human blood types. Last two things I want to talk about are something called pleiotropy and polygenic inheritance. Now, these are not concepts we're really going to do problems with, or like dihybrid crosses, or look at individual probabilities, but they're more just something I want you to understand. So pleiotropy is when one allele leads to multiple traits. So for instance, you could have an allele that produces a protein that ends up in your skin and in your small intestine. And that protein does different jobs in each place. But what that means is inside your DNA, that's one protein that must be produced. And that one protein can give you multiple traits. And that's actually one of the ways that living organisms are able to be so complex. Because you can take that genetic information you have and basically multi-purpose it. Now, the opposite side of that is what we call polygenic inheritance, where you have multiple alleles that lead to one trait. And that's actually how you end up with the complexity that is human height or human skin tone. Because there's like 10 or 15 different alleles that combine to give you a variety of possible skin tones, or that combine to give you a variety